Good day to everyone. So this is a lecture video about bioanalysis. Okay, so basically when we say bioanalysis, we're simply, let's try to break it down into two in its component words. No? So this is a compound word comprising of bio and analysis. So when we say bio, we are referring to life. Anything that is biological, anything that has life is considered to be part of our biosphere. And we all know what analysis means. So when we say analysis, simply means that we are trying to analyze the components of something. So in this case, bioanalysis means that we're analyzing the components of life. So let's start with the basics. Now, the living matter is composed of mainly six elements, and this is something that we have to understand. We have carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Now, these elements together constitute about 90% of the dry weight of the human body, some more than others, some less. Okay, so like for instance, you have hydrogen, which is, well, the most abundant element in our universe. You have carbon, which means that we are carbon-based life forms. You have oxygen, which we breathe in, and you have nitrogen, phosphorus, and sulfur, mainly for the proteins in our body. But altogether, these biomolecules comprise about 90% of the dry weight of the human body. Now, bioanalysis may be defined as the laboratory analysis of biomolecules, and biomolecules are organic compounds with biological activity, generally important only in biological systems or what we know as cells, organs, tissues, and so on. And biochemistry is the study of the structure and the function of these biomolecules. And as a related concept, we have biotechnology, which concerns itself with the industrial applications of biochemical techniques. So we have lots of descriptions, but all in all, these are just, well, it's base component, analysis, molecules, chemistry, and technology, all added and applied to life forms. Now, there are four groups of biomolecules. You have carbohydrates, you have lipids or yung fats, you have proteins and nucleic acids. And all of these four are essential to the life of human beings and all carbon-based life forms and are therefore found in all living cells. And we refer to them as organic compounds. Now, based on the element carbon, all four also contain hydrogen and oxygen. So we are carbon-based life forms, and carbon is a four-valence element, and thus sometimes they don't necessarily form bonds with larger molecules, and they use hydrogen and oxygen to terminate their bonds. Okay. And you have proteins, nucleic acids, and a few lipids contain nitrogen. And of the four, proteins alone contain sulfur, while nucleic acids have and some lipids also contain phosphorus. And the two most important biomolecules in modern bioanalysis are proteins and nucleic acids, and you'll find out why in a few slides more. Now, organic compounds are about 25 to 30% of the cell weight. These are nucleic acids, proteins, polysaccharides, sugars, and lipids. Now, proteins account for about 10 to 20% of the weight of the cell. Nucleic acids about 7 to 10%. Polysaccharides or carbohydrates, or more commonly known as sugars, account for about 2 to 5% of the cell weight. And 3% of the cell weight is due to lipids or fat cells or fat. Okay, but obviously fat cells, they contain more lipids. So these percentages may vary depending on what kind of cell we're talking about. And proteins account for more of the cell weight in cells like erythrocytes. But generally, proteins are, well, larger molecules. They are heavier, so they comprise about or the largest weight of our cells. Okay, first, let's talk about carbohydrates. Carbohydrates, which include sugars and starches, contain carbon, hydrogen, and oxygen. Perhaps the simplest biomolecule we can muster are or is carbohydrates. So it's just comprised of carbons, hydrogens, and oxygens. And they are classified into three types depending on the size. When I say size, I'm referring to the length of the chain. So you have monosaccharides, which are, as the term suggests, single unit sugars. You have disaccharides, which are double unit sugars. And you have polysaccharides, which are longer chains, so more than 2 units of sugar. Now, carbohydrates, they provide a ready and easily used food source for cells. That's the reason why when you eat sugar, you are a lot more energetic. Okay, that's also the reason why when we eat lots of food, we typically get sleepy. It's because our body requires energy to break down that energy and convert it to more energy. So our body is suggesting a more efficient way to conserve energy by reducing all the different supplies of energy all throughout our body and focusing on digestion. Okay, so those are the basics of carbohydrates. Now, monosaccharides are the smallest carbohydrates, and common examples of those are aldoses and glucose, galactose and ribose. You also have ribulose and fructose, which are ketoses. So, all of these are single unit sugars, and they are classified according to the number of carbon atoms in their chemical backbones. So, for instance, a 3 carbon monosaccharide is called a triose, as it has 3 units of carbon, and it is triose. And a 5 carbon is pentose, because pent translates to 5, and a 6 carbon, 6 translates to hex, so it's referred to as hexose. Now, glucose, galactose, and fructose are hexoses, while ribose and ribulose are pentoses. Now, these monosaccharides, glucose, galactose, and fructose, ribose, and ribulose. So these are the important biomolecules that are typically involved in our citric acid cycle or our pentose monophosphate shunt, basically all the different branches of biochemistry where the human body produces ATP, or in other words, energy. So these are the relevant sugars that you have to pay attention to. Okay. Now, in nature, monosaccharide exists in two forms, rings and open chain carbon skeletons. And we'll be discussing that in our next slide. Okay. So, based on the number of carbon atoms, here is an illustration of the chemical compositions or the chemical formula of each of these aforementioned monosaccharides. Now, moving on to disaccharides, they consist of any two monosaccharides covalently linked to each other. So, a common example would be sucrose. You also have a very familiar lactose, which is the main sugar found in milk. Excuse me. And maltose, which is just glucose and glucose. So, typically found in whole grain or grain sugars. So, these are natural. They tend to be exclusively either plant or animal products. Like, for example, sucrose is what we know commonly as table sugar. And maltose is, again, from grains. And then you have lactose, which is found mainly in milk. Now, the largest and most complex carbohydrates are the polysaccharides. They are polymers, long chains of repeating chemical units. And the examples of these are plants, polysaccharides from plants, which are starches, which is very important in the diet of humans. We also have cellulose in plant cell walls. So cellulose comprise mainly the covering of corn. So if you're familiar, if you're familiar with how corn usually goes through our body, so we digest a small portion of the corn, or if it's mashed, then we can digest it. But the covering or the wall of the corn, the actual crunchy part, is actually indigestible. It's made mainly of cellulose, and cellulose is something that we cannot digest because we lack the enzyme cellulase. Okay, and this is beneficial for our diet because it helps our intestinal motility. Next, we have lipids. So lipids are all hydrophobic, meaning they are water-fearing. So that's the reason why when you wash the dishes and you use a lot of oil, or if you extract a lot of oil from the food you're cooking, it's a bit hard to clean the dishes because lipids, they don't mix with water. Okay, so they push water away, and when the lipids coat the plates, the pans, or any kind of cookware, the water tends to just slide down. Okay, most lipids again are insoluble in water but readily dissolve in other lipids or organic solvents. So lipids are soluble in alcohol, they are soluble in hexane, they are soluble in benzene, they are soluble in a lot of organic solvents. And then the most important or the most abundant lipid in the body are triglycerides, phospholipids, and steroids. Now, triglycerides is something that we're very familiar about because we've recently been warned about the dangers of eating too fatty foods or eating too much, which causes obesity and therefore increases triglycerides, which can clog up our arteries. Okay, so that we're very familiar with, but phospholipids and steroids are a bit more obscure to our knowledge, so we'll discuss it later. But anyways, try acylglycerols or triglycerides. These are fats and oils which consist of three fatty acids linked to a molecule of glycerol. So a three-carbon alcohol. Fatty acids are a long carbon chain molecule, each with a single carboxyl functional group. Now, in nature, triacylglycerols are widespread and are crucial energy storage molecules, and they function as insulation in mammals. So, this is the way our energy is stored. This is the fat in our bodies. Now, triacylglycerols. Now, phospholipids have a hydrophilic and hydrophobic region. It is present in cell membranes and allows cells for what we call selective permeability. Um, which allows the cells to select what may enter or leave. Okay. And we have steroids as well, which are common lipids without fatty acids. So chemically, they consist of a fused carbon ring consisting of four rings, three rings of which are six-membered and one which is five-membered. So when we say a six-membered ring, we're talking about the carbon atom. So you have one, two, three, four, five, six, and then you have one, two, three, four, five. Again, you have three six-member rings and one five-member ring. Okay. Now, some steroids such as cortisone function as hormones. Cholesterol is an important component of membranes in animal cells. So these are a few examples of where we can find, excuse me, steroids in our body. Next, we have proteins. Now, proteins account for over 50% of the organic matter in the body, and they have the most varied functions of organic molecules. Now, proteins, again, they have a very, very, very wide range of functions in our body, but the main function they have is that the building blocks of our smaller molecules, or rather, they are the building blocks of our bodies, and they are built by what we call amino acids. So think of it as, let's say, if you have monosaccharides for carbohydrates, you have your glucose, you have your galactose, and so on. For proteins, we refer to them as amino acids. So these amino acids are the building blocks or the smaller molecules that make up longer proteins. Now, amino acids are joined together in chains to form large, complex protein molecules. Now, they also have configurations, like, for example, insulin is in its primary configuration. You also have secondary configurations, tertiary and quaternary configurations, like hemoglobin. Okay, but those are for later and more complex discussions, more in-depth discussions about proteins. For now, just a short introduction of what proteins play in our body. Okay, now they are large polymers with intricate three-dimensional structures and tremendous diversity. So once they exhibit their three-dimensional structures, they are referred to as tertiary proteins. Now, these proteins, these complex molecules, are built by, again, amino acids, which are monomer units, or which are its monomer units. And historically, amino acid sequences have been used to trace possible evolutionary relationships among organisms since they are coded by DNA-based sequences. Now, our DNA determines how we make proteins. Okay? Mutations, therefore, are manifested structurally by different amino acid sequences of proteins. Now, these are our amino acids. So you have glycine, alanine, valine, isoleucine, leucine, phenylalanine, tyrosine, tryptophan, lysine, arginine, and so on. Okay, so these are the building blocks. These are amino acids that build up our proteins. So peptides are short chains of amino acids linked by peptide bonds, and most biologically active peptides contain 2 to 10 amino acids. So these are like your insulin proteins. So you have also aspartame, which is a dipeptide. It's comprised of aspartic acid and phenylalanine, which is the artificial sweetener marketed under the trade name NutraSweet. You also have glutathione, a tripeptide, which is an antioxidant believed to be an anti-cancer agent. You also have enkephalins, which are found in analgesics. Okay, so analgesics are fever relief medicine. And you have oxytocin, which is a hormone composed of 9 amino acids, which induces labor by stimulating contraction of the uterine muscles. So these are a few examples of the proteins that are relevant to the human body. Okay, we have other proteins depending on our their function in our body. So you have fibrous or structural proteins, which are strand-like proteins that appear most often in body structures. So they're important in binding structures together. A good example of this would be your collagen. So collagen is a protein. So I'm sure you're very familiar with how collagen is used in the beauty industry right now. But it is actually able to supplement the tautness of our skin. So it prevents sort of wrinkles and so on. It keeps our circulatory system intact, meaning that it keeps our veins, our arteries from collapsing. It keeps them strong, and it also helps in our heart. Okay. We also have globular or function proteins. So these are mobile, generally spherical molecules that play crucial roles in virtually every biological process we have. So some help with immunity, others help with growth and development, and others help us digest the different nutrients that we intake in our body, and those are our enzymes. Okay, enzymes are functional proteins that act as biological catalysts. So when we say biological catalysts, these are substances that help the reaction go faster. Okay, it helps it go faster. It helps it get more efficient, and at the same time, they don't change the product itself. Okay, so they help the reactants of a chemical reaction occur without affecting the product itself. So these are catalysts, and these catalysts are very important to life in such a manner that if they were to disappear, we will not be able to sustain life. The reactions are going to be too slow for us to sustain life. Now, enzymes are very specific in their activities, each only controlling only one or a group of chemical reactions and acting only on specific molecules. So they are not allowed, or rather, they cannot actually react with molecules that they're not designed to react with. They're very specific. Okay, next we have nucleic acids. Nucleic acids are the building blocks of our DNA and RNA. So basically, these are our genetic fingerprint. So like proteins, nucleic acids are polymers with nucleotides being the monomer unit. So if proteins have amino acids, nucleic acids have nucleotides. Okay. Now, the role of the DNA is without question very fundamental to the blueprint of life, meaning that without DNA, without RNA, we are not able to exist. Okay. They determine our physical appearance, they determine our mental resilience, they determine everything about the human body or the body of an organism that it is producing proteins for. Now, DNA is the genetic material found in the cell of the nucleus. So, or in the cell nucleus. Now, it has two roles. One is to replicate itself before a cell divides, so as to carry on its instructions. And second, it provides instructions for building every protein in the body. Now, we also have RNA and many different types of RNA, but again, we'll go more in depth for that in a more in-depth lecture about nucleic acids. Okay? It is considered as a molecular slave of DNA. It is because RNA carries out orders for protein synthesis issued by DNA. So DNA is the actual instructions. RNA is the one that executes that action or that instruction. Okay. So these are again the major complex biomolecules of cells. You have protein, deoxyribonucleic acid or DNA. You have ribonucleic acid, RNA. You have polysaccharides and lipids. Now, in the laboratory, we of course have to be able to analyze these biomolecules. Now, electrophoresis, as mentioned in our previous lectures, is a group of techniques used to separate and study molecules using electrical charges. Now, based upon these charges, both sign and magnitude, biomolecules such as proteins, peptides, amino acids, nucleic acids, and fragmented nucleic acids migrate in an electrical field. Now, if you'll notice, electrophoresis is very useful for DNA, RNA, and protein analysis, but not very much useful for neutral molecules like lipids or um, carbohydrates. Now, matrices for separation include paper, various gels, and liquids. Now, in paper electrophoresis, we use an absorbent filter paper soaked in current-carrying buffer, and this type of electrophoresis is commonly applied to amino acids and peptides. And we also have gels. We can use polyacrylamide gel and agarose gel. I think agarose is a lot more common in our practice. Okay. Now, page or polyacrylamide gel electrophoresis is usually run vertically, while agarose gels are horizontal. Now, PAGE is commonly used with proteins, while agarose gel electrophoresis is a favored method for nucleic acids and their fragments, but it is also effective for proteins. Now, in capillary electrophoresis, separation occurs in a glass capillary tube containing the buffer only. Next, we also have chromatography. Chromatographic techniques are also powerful tools for the analysis of biomolecules. Various types of chromatography are useful, especially for analyzing proteins and nucleic acids. Now, we have paper chromatography, which is very useful for separating amino acids found in food samples. Now, if you remember in our chromatography lecture, we have different types of chromatography based on its interaction. We've also discussed the different interactions of chromatography. So basically, all I want you to know is that this is good for separating amino acids found in food samples. Again, thin layer chromatography is a similar application to paper chromatography, but this time we have what we call a TLC plate. Okay, components may be separated based on the differences in molecular weight, charge, or polarity. You also have spectroscopy. This is the method preferred or used in most clinical laboratories, if not all. So you have various spectroscopic methods in the laboratory, and we have different wavelengths for each of the biomolecules that we're trying to examine. Okay. Now, all proteins have some UV absorption at 280 nanometers, and all nucleic acids absorb at 260 nanometers. So these are the ideal wavelengths to which we can analyze proteins. We can also use immunoassays. Immunoassays use antibodies, which are complex proteins directly involved in the immune response against foreign objects, such as viruses and bacteria. So they are produced by the immune system in response to the entry of anything foreign to our bodies. Okay, similar to proteins, antibodies and the products of the reaction of antibodies with drugs absorb UV and visible light. Their presence in blood, urine, semen, and other body fluids may be detected by this method.